introduce myself. I'm Andrew Carroll, Head of Science at DNAnexus. I'll just be giving a brief introduction to what DNAnexus is and how we work with Sention. And joining me is Don Freed, who will be doing most of this presentation, who is one of the lead bioinformatics scientists at Sention. So DNAnexus is a cloud-based platform which serves as a network for computational biology, precision medicine, bioinformatics, through which members can collaborate with each other and with scientists at DNAnexus, regardless of where they are throughout the world. The company today, we're around almost 100 people or so. We have multiple offices in the United States, in UK, in Asia as well. We have many enterprise customers. We have hundreds of thousands of individual projects that are collaboratively shared on the platform. We have more than 10,000 users across 30 different countries in the world, widely used by our customer base in pharma R&D, and in government, in NIH consortia, such as the IMERGE consortia and the CHARGE consortia and the ENCODE consortium, as well as the backing for the Precision FDA platform. We are multi-cloud. We can run on Amazon and Azure. We can also run on multiple regions throughout the world, so that we can keep your data. If it needs to stay in say, the EU, it can be in Frankfurt. If it needs to stay in Australia, it can be in Australia or Singapore or in the United States, and presents it all with a single common pane of glass, so that you have a uniform development experience. The platform itself is built on a number of foundational underlying technologies. One of the first of them is security and compliance. So we've invested a great deal in having a very highly secure platform. This has been vetted by the Department of Health and Human Services, has given authority to operate, is the platform for Precision FDA. It's compliant, so you can run clinical pipelines. We have a number of different engagements which run CAP, CLIA, compliant pipelines. We can operate in a HIPAA compliant manner. And a couple of the things which this particular collaboration uses is our ability to give access to tools and software on the platform. So we work with partners, for example, Sention, to put their bioinformatics into the platform so that you can deploy it in your operations. You can run it in a very streamlined and smooth manner. You can have full data provenance and tracking over the analysis that's been done. And the licensing is all managed. So you don't have to worry about large upfront licenses. You can pay as you go, and you can just run these as any other application and essentially receive the entire benefit of a technology like Sention. The last slide that I'm going to talk through is just to give some framing for why players like Sention and DNAnexus are going to be important in the coming years. I think that everybody in this space has recognized that the amount of genomic sequencing is increasing exponentially. Just 3 or 4 years ago, having a 10,000 exome project was an enormous deal. Not many places had that. And now people are talking about hundreds of thousands of genomes as the standard project that will be executed. And when you look at the total potential volume of data in 2015, there was an analysis conducted that if we sequence a reasonable fraction of the world's population, we'll have data which dwarfs a number of the largest repositories of data that currently exist. And the sort of efficiency with which you would operate when you're operating at small scale is different than that which is required to operate on hundreds of millions of genomes. And that's one of the reasons why we believe that companies like Sention, which are producing optimized technology to more efficiently look at this data, will be important in being able to scale to this volume, and why platforms like DNAnexus, which help provide access to those tools and help you collaborate on that large-scale data, will be foundational to the way people will operate on genomic data in the future. And with that, I'm going to hand the presentation off to Don, who's going to talk through the implementation of Sention and DNAnexus with some specific use cases and examples.
Okay, so thank you, Andrew. I'll just start off my talk by saying that at Sention, we're very, very excited to have the Sention tools on the DNAnexus platform. We think that the combination of the Sention tools and the DNAnexus platform essentially provide the best possible solution for processing large-scale genomic data sets. So we're very, very happy to be a part of this. I'll just start off my talk with a brief introduction to de novo mutations. We'll talk about what de novo mutations are, how they're defined, and their impact on population and clinical genetics. From there, I'll move on to talking about Sention. I'll give a brief overview of Sention as a company, and then we'll spend quite a bit of time talking about the Sention genomic tools. Then we'll look a little bit under the hood of the Sention Rapid DNA-seq app. We'll figure out how it works to process a whole human genome in about 1 hour. And finally, we'll end the talk with a brief tutorial of how to analyze data using the Sention Rapid DNA-seq app. So great, let's jump right in. So here's a brief introduction to de novo mutations. De novo mutations are defined as mutations which are identified in individuals, but not that individual's parents. Every human has about 50 to 100 de novo SNVs and indels, which are not present in their parents. Because these mutations are not inherited, they seem to appear out of nowhere, which is how they get their name de novo, which means from nothing. De novo mutations can be any type of genetic variation, not just SNVs and indels. They can also be structural variants, copy number alterations, and other sorts of mutations. De novo mutations are the primary source of genetic diversity in the human population, which geneticists typically think of as being a very good thing. So genetic diversity allows the population to be resilient to disease and resistant to viral and bacterial infection. So this is a picture of what geneticists are typically thinking of when they think of a de novo mutation. Here we have a pedigree with mom and dad on top and 5 male children down here on the bottom. Underneath the symbol for each individual, you can see a Sanger sequencing trace of the locus. As you can see, both mom and dad are homozygous for the A allele at these sites, and so are 4 of the 5 male children. However, one of the male children is heterozygous for a C allele at the site of interest, indicating that C allele must have arisen de novo in that child because it's absent from either that individual's parents. So as I mentioned earlier, genetic diversity is typically thought of as a great thing by geneticists. However, genetic diversity does have some downsides, and the downsides are essentially genetic disease. So many very severe genetic diseases frequently occur as a result of de novo mutations. Some examples are Rett syndrome and very severe intellectual disability. When you have a case where an individual has a genetic disease that's a result of a de novo mutation, having genetic information from other members of the pedigree, especially parents, can be crucial for actually honing in and identifying the causal genetic variant. So this brings us to applications of rapid DNA sequencing in the clinic. So every year in the United States, you have newborns who are born with life-threatening conditions that are a result of Mendelian genetic diseases. Overall, across all infants who are born in the United States, this is actually relatively rare, but these Mendelian genetic diseases actually account for a significant fraction of infant deaths. So rapid DNA sequencing of these critically ill infants has been shown to actually potentially improve outcomes of some patients. So rapid DNA sequencing has a high diagnostic yield. For some cohorts of very ill children, it can be up to 73%. And then it's also shown to have a beneficial impact on outcome for about 23% of these patients. So personally, as a scientist, I think this is really cool that, just 15 years after the sequencing of the first full human genome, we're actually able to use rapid DNA sequencing of newborns to actually improve their clinical outcomes, which I think is just amazing. So now I'll move on to talking about Sention. So at Sention, our mission is that we want to enable precision genomics data for precision medicine. What that means is we want to enable our users to process their big genomics data at an affordable cost and also in a reasonable amount of time. When our users process their genomics data with Sention, we want them to have confidence in the results, and that confidence will both come from the high accuracy that our tools provide and the fact that our tools provide consistent results every single time that they're run. So Sention provides this to our users through our high-performance analytical software. So when we think about high-performance analytical software, we typically think of that analytical software as having 3 components. The first component is the mathematical models. The second component is the computational algorithms. And the last component is the software implementation. So the Sention tools use the exact same mathematical model that's used by the tools produced by the Broad Institute. This really provides Sention and our users two really big benefits. The first benefit is that our tools provide identical results to the tools produced by the Broad Institute within the run-to-run variation of the Broad Institute's tools. So what that means is that users can immediately switch from the Broad Institute's tools to the Sention tools and have confidence that they'll be seeing essentially the same results. Another big benefit that having the identical mathematical model provides to our users is that our users can immediately benefit from the years and years of work that the Broad Institute has put into developing and optimizing their mathematical model, which is really a great thing. So the next step of the next portion of the analytical software is actually the computational algorithms. This is an area where Sention's team can bring their innovations to our bioinformatics software. So Sention's team has developed improved computational algorithms to produce the exact same identical results as the Broad Institute's computational algorithms, but with greatly improved runtime performance. Because of this, the Sention tools are able to process genomic data at any depth without need for downsampling or throwing away some reads. And then finally, the last component of the analytical software is the actual software implementation. Sention benefits from an enterprise-strength software implementation. The developers who have developed the Sention software have decades of experience developing enterprise-strength software. Each release of the Sention software goes through a very rigorous quality assurance process, which guarantees that the number of bugs in our software are minimized. And finally, because we have the enterprise-strength software implementation, our software is typically more robust for many different applications. So this is kind of the approach that Sention has taken. So we're sticking with the software approach, but we're using improved computational algorithms and better software implementation to increase our runtime performance. So the results of that work really speak for themselves. Shown here on this slide is the runtime of the GATK 3.5 versus the Sention equivalent pipeline. This is going from a lined NAND file to a VCF file. On the x-axis, we have whole genome sequence samples, starting with 5x coverage samples on the left, all the way up to 51x coverage samples on the right. And as you can see for yourself, the Sention tools provide a drastic performance improvement relative to the GATK pipeline. The Sention tools offer a number of benefits beyond speed. Again, as I mentioned earlier, our tools prefer no downsampling and have no thread dependency. So every time that you're going to be running the Sention tools, the results are going to be exactly identical. This actually results in slightly higher accuracy for our tools than the Broad Institute's tools. Because the Broad Institute's tools are performing downsampling, they're throwing away some data. And what can happen is if you happen to throw away the wrong set of reads, you can potentially end up with false positives or false negatives in your call set. As I mentioned earlier, the Sention tools are very robust. This enables our tools to perform joint genotyping on over 100,000 samples simultaneously. And with our partnership with DNAnexus, our tools are very easy to use. They're available today on DNAnexus, and you can run them in both regular and rapid run mode. So I've just spent a lot of time talking about what I think are the benefits of the Sention tools. Thankfully, you don't have to take my word for it completely. The Sention team has participated in a number of third-party independent benchmarks, which have really validated the strength of the Sention's approach. So one of the most prominent third-party challenges that the Sention team has participated in are the Precision FDA Awards. There's been 2 Precision FDA Awards which Sention has participated in. The first is the Precision FDA Consistency Challenge, and the second is the Precision FDA Truth Challenge. How the Precision FDA Consistency Challenge works is that the FDA set out to evaluate whether bioinformatics software was able to process a single sample consistently, and again, that's deterministically. As I mentioned earlier, this just simply validated that the Sention tools do in fact process the data, resulting in the same set of variants every time that our tools process the sample. And the other aspect of the consistency challenge is different and perhaps a little more interesting. What the FDA set out to do was they took a single biological sample and then processed it through 2 different library preparation methods and set out to evaluate what was the consistency of the variant calling pipelines after those two different library preparation methods. Again, the Sention tools performed very well here. They actually had the highest consistency of any of the tools in the challenge, and because of that, the Sention tools were awarded the highest overall performance award and the highest reproducibility award in the Precision FDA Consistency Challenge. The other Precision FDA challenge that the Sention team participated in is the Precision FDA Truth Challenge. In this challenge, the good people at NIST developed a truth set for a sample that was highly validated, but they did not release the truth set to the public. So with this hidden truth set, they asked for submissions after processing some genomics data, and then they evaluated the accuracy of the submissions relative to the hidden truth set. With the Precision FDA Truth Challenge, there was no overall award winner, but the Sention tools were awarded the highest indel precision and the highest SNP recall awards. So I'll dig in a little more to the Precision FDA Truth Challenge results so we can really explore the value provided by the Sention tools. And this is a big table which shows the Precision FDA Truth Challenge results. The sample HG002 is the sample with the hidden truth set that was not publicly released. The sample HG001 is a sample with a publicly available truth set before the challenge occurred. So as you can see, the Sention genomic tools are in the top row, and the Sention genomic tools provided very balanced accuracy across both SNPs and indels for both samples. So that indicates that the Sention model was not overly trained to one sample or another. So we like to use this slide to make the argument that if there was an overall award given for the Precision FDA Truth Challenge, the Sention team should at least be in the running for that award. And finally, it's not the focus of today's talk, but Sention also has another product line called Sention TNseq. Sention TNseq provides identical results to the Broad Institute's somatic variant discovery workflow with the Mutect or Mutect2 variant callers. So the Sention TNseq provides an end-to-end solution for processing your genomic data, starting with FASTQ and then going all the way to VCF files. Again, Sention TNseq benefits from kind of the same benefits that are provided by Sention DNAseq. It benefits from the improved computational algorithms and the more robust software implementation, resulting in about a 10x performance improvement relative to the standard Broad Institute pipeline. The results here are, again, deterministic and are very robust to different types of genomic inputs. So now I just am going to move on to looking under the hood of the Sention Rapid DNAseq app. So it's important to remember that many types of genomic analyses are nearly perfectly parallelizable. And what I mean by that is that you can divide your data set into chunks or divide your genome into different regions, process those regions independently, and still end up with the exact same results if you're careful about how you do this. So some steps of the analysis pipeline that are very parallelizable are, for example, alignment to the reference genome, some of the pre-processing steps, including indel realignment, and finally variant calling. And again, what I mean by perfectly parallelizable is that these steps of the analysis pipeline can be completed independently on different parts of the data. The Sention tools are built with this sort of distributed processing in mind. So what that means is that the Sention tools are going to provide the exact same results, whether they're being run on one machine or many machines simultaneously. And what the trick is behind the actual Sention DNAseq Rapid Run app is that the work is actually being distributed across multiple servers simultaneously to decrease the total runtime. So this is a picture of the standard Sention DNAseq pipeline. Up at top, we have our raw sequence reads, and then they're going through the steps of alignment with the Sention BWA, duplicate marking with Sention locus collector and Sention dedupe, indel realignment with the Sention realigner, and base qualities for recalibration with Sention qualcal. And finally, the last step of the pipeline is variant calling using the Sention haplotyper. So again, this is what the distributed pipeline looks like. Again, our users are bringing their raw sample reads to the DNAnexus platform and then using the Sention app to process the data. So you can think of each vertical column here as one machine, And again, the work of this genomics pipeline is being distributed across many machines, which are processing the data in parallel to provide the speed up. So the results here are the same as the results of the linear pipeline. However, because we're using so many different machines to process the data, again, we get a very significant performance increase. So now I'll move on to the tutorial where we'll actually show you how to analyze some data with the Sention Rapid DNA-seq app. We'll go through the steps of registering for the Sention Rapid DNA-seq app, uploading some samples to the DNAnexus cloud, and then running the app using the DNAnexus graphical user interface. So you can get started using the app today. Go over to www.DNAnexus.com/Sention, and you can sign up, and you'll be given immediate access to the app. So you can get started using the app very quickly. Once you've got access to the app on the DNAnexus platform, the next step is to actually upload some data. You can upload your data in different ways. So the DNAnexus platform allows you to actually drag and drop files from your local computer, which is very nice. You can upload files from a server, or you can transfer them over from another project on DNAnexus. Another way which you can upload data is through the DNAnexus command line tools. I found the DNAnexus command line tools very easy to use. I was pleasantly surprised. So all you have to do is download and install the tools, log in, and then use the dx upload command to actually upload some data to the DNAnexus platform. And then finally, the last step is actually running the app. To run the app, you have to select the Sention FastQ to VCF app, the rapid version, from the list of apps that you have on DNAnexus. And then once you find the app, you need to configure your workflow to process the data. So shown on the left here are different input samples. So in this example, we have 3 different input samples processing the data. My cursor is a little tiny, but this is one sample. Here's another sample, and this is the last sample. All of these samples are being input to the Sention FastQ to VCF Rapid Mode app. And the results of running the Sention FastQ to VCF Rapid Mode app are variant calls in GVCF file format. These variant calls in GVCF file format are all being fed into the Sention genotyper, or GVCF typer, which is going to produce a VCF file containing variant calls across all samples simultaneously. Because you're running this app on the DNAnexus, this scales with the cloud. So what that means is you can process thousands of samples simultaneously and scale the analysis to thousands of machines. So even if you're running thousands of samples all at once, the total processing time for all samples should be approximately 1 hour. The GVCF typer, the Sention GVCF typer on the DNAnexus platform also scales to thousands of samples without the need for intermediate file merging. So that means you can take all the GVCFs that you have, input them to the Sention GVCF typer, and you get out a VCF file containing all the variants in all of your samples. So scientifically speaking, the last kind of step of the pipeline is the actual identification of de novo variants if you have a trio sample. So it's very common for many academic papers that the papers end up identifying de novo variants using some hard filters. So some of the most common hard filters are ensuring that the variant does not have any support in the parental sample. This is important because it can remove potential mosaic variants that are occurring in the parental sample. Ensuring that the variants are not present in any unrelated individuals who might be in your cohort. Ensuring that these potential de novo variants are not present in dbSNP or the Nomad databases. Again, this it is going to potentially remove many false positive de novo variant calls. And then finally, ensuring that the de novo variant is supported by at least a minimum number of reads. So I'm not going to give out any definite recommendations here because you're going to have to tune this based on your preferred level of sensitivity and specificity. But this is a very common approach for identification of de novo variants. Something else you can do is a more sophisticated approach where you use a Bayesian model to input your prior expectation of finding a de novo variant at a particular site. This is a little more complicated, and we won't cover exactly how to do that here. So in conclusions, the Sention tools on DNAnexus platform provide an excellent solution for processing your genomic data. They're very easy to use. The software is already installed on DNAnexus, and it's ready to go. You just head over to the website, which I mentioned previously, and we'll mention again, and you can get up and running with the Sention Rapid DNA-seq app in minutes. I think the standard Sention DNA-seq app is able to process your genomic data about 10x faster and with a 5 to 7x reduction in expenses relative to the standard BWA GHK pipeline on DNAnexus. And again, the Sention tools are 100% consistent and have no run-to-run variation. So every time that you're going to be running the tool, it's going to be producing the same result. Joint genotyping is built into the Sention tools with the Sention GBCF typer, allowing you to jointly call variants in thousands of samples simultaneously. So this really works well with the strengths of the DNAnexus platform. So DNAnexus is able to, so let me just put it this way, the Sention tools are really bare bone tools. They just are, binary tools for processing genomics data. However, this works really well with the DNAnexus platform, which offers a integrated platform solution for processing your genomics data. Perhaps most importantly, the DNAnexus platform can provide great data security, great data provenance, reliable and collaborative environment in which to process your genomics data. So finally, I'll just mention that both DNAnexus are going to be at BioIT World next week. And Brendan Gallagher from Sention will actually be at the DNAnexus booth next Wednesday from 3:30 to 4 p.m. So if you would like to talk to someone from Sention in person, please stop on by. So I think that's the end of my talk. And now we'll have a brief question and answer session.
Yeah, but before I'll go through the questions that have been